Hello, my name is James Guest. I'm a professor of neurosurgery at the University of Miami in the Miami Project to Cure Paralysis. And today I want to talk about lessons from the Miami Project Schwann cell transplant programs for spinal cord injury. And I'd like to make you aware of the advantages of regenerative repair as compared to some other therapeutics, for example, neuromodulation. So with regenerative repair, we're really getting restoration of circuits. There's a potential for multi-target repair, and we can achieve long-term stability of the outcomes. Furthermore, we're using natural signal processing through circuit restoration, and in some cases, we have a reduction of maladaptive plasticity, which is one of the most difficult consequences of living with spinal cord injury. So many types of tissues and cells have been transplanted for spinal cord injury over a period of 40 or 50 years now. And these started with fetal and embryonic tissues, peripheral nerve grafts, Schwann cells, and then into the neural stem cell era with oligo precursors and then neural precursors, and most recently induced pluripotency neural stem cells. Just because something is recent doesn't necessarily mean that it's better. So we take heart from the fact that regeneration of the spinal cord is actually possible. So this is an old paper from the Newt where the spinal cord was actually resected. And then 40 days later, you can see restoration of the gray matter and white matter of the spinal cord in this type of animal. And so that tells us that it's biologically possible to repair the spinal cord. There are a lot of unresolved issues in regenerative transplantation, one of which we find in our clinical programs is getting optimal cell survival, but also, importantly, integration of the graft that we place into the CNS effectively. Furthermore, the timing that can be subacute or chronic has not fully been optimized. I want to talk a little bit about the importance of peripheral and central tissue compartments on regenerative potential. So I'm going to use this figure that was given to me by Dr. Kukula several years ago to just walk you through what a human spinal cord injury looks like in the chronic phase. So we can see gliosis over here near my pointer, which is intense scarring. We can also see that the cavitation in the spinal cord is very complex. And I'll be making the argument that these spaces are actually outside of the central nervous system. Furthermore, we can see that the spinal cord is tethered to the dura here. And there's no gray matter because at spinal cord injury epicenters, typically the gray matter is lost. So this is what we're actually working with in many spinal cord injury situations. So one of the key things to think about is what is called the glial limiting membrane. The glial limiting membrane is like a wall that seals off the brain and the spinal cord from outside, from external cells, from proteins, and it's created by the foot processes and astrocytes that link together in a very tight boundary. And that's also shown over here in this electron micrograph where we see axons and then astrocyte processes. And then we're outside of this tissue when we can see all of this basal lamina. And so the glial limiting membrane is always damaged in spinal cord injury, and it's always part of the wound healing process. One of the most useful ways to think about this is to look at what is called the dorsal root entry zone. This is where sensory fibers grow into the spinal cord and connect in circuits. And what I'm showing you here is what is called a freeze fracture study. So what they did here is they took the root entry zone and they split it apart like a book. So we can see over here the peripheral axons, which are bundled inside connective tissue compartments. And then we see on the central face, which is the spinal cord side, a lot of abundant astrocyte processes and no visible pores for the axons to enter into the central nervous system, except very occasionally, as I've indicated down here. So this kind of boundary is recapitulated after spinal cord injury. I'd like now to talk about residual tissue bridges. So when the spinal cord is injured, and I've shown here a pre and post decompression MRI, you can see the signal change in the spinal cord. This tissue is going to be lost and not replaced in the spinal cord. It's going to evolve to what we call a tissue cavity. Now, when we look around the cavity, we can see preserved strands. So this is a T2 MRI study. And so we can see that these strands can be measured in millimeters, and they're beginning to serve as a biomarker of the preservation of the spinal cord that's associated with outcomes. And when we think about tissue bridges, we can consider that when there are extensive tissue bridges, we expect recovery, and therapies that would amplify neuroplasticity are good for this type of situation. However, when we have no tissue bridges, we will not be able to engender much neuroplasticity, and we need to replace structure to achieve connectivity. And I've shown that again here. This is a hemisection injury, but what I wanted to indicate is what can happen with neuroplasticity and what can happen with a transplant repair. So in neuroplasticity, if we can effectively engender new process growth, that can be around the injury and grow to target neurons. However, if we want to replace the tissue, then we're trying to achieve actual regeneration of the cut processes. And those cut processes can grow through our graft and form a new connection on the neuron that then goes to important target neurons like those for muscle, those for the bladder. So again, this concept that we're inside the injury, we are outside the spinal cord. And now I'm going to talk about the advantages of using Schwann cells for therapeutic repair. They are very trophic cells, and they engender tissue repair effects like neovascularization. They create a lot of basal lamina. They can remyelinate axons, and they can promote axon regeneration on their surfaces. Here's what the differentiated Schwann cells look like. So this is a classical myelinated figure for high-speed axonal conduction, and then these are ensheathed small axons. Here's what happens when we have a peripheral nerve injury, and this is what we want to have happen also when we implant the cells into the central nervous system. Here we see that the Schwann cells have no axons with them. They form these structures called bands of Bunger, which are highly regenerative pathways that axons can grow into. And these pathways are always encircled in compartments. Remember what I just showed you about the compartmentalization in the peripheral part of the root intrazone. It's the same with these types of Schwann cell implants. So let's talk a little bit about myelin loss and replacement. So after spinal cord injury, myelinating cells are lost, axons may be preserved, but remyelination may not occur fully. And so now I want to talk again about the tissue bridge concept, because what's actually happening when we have compression or contusion injuries to the spinal cord is the axons are spared on the outside, but they're lost on the cavity side, which I'm indicating here. And now there's a gradient of injury. And in that gradient of injury, we get different cells that are responding and effecting the repair. These are demyelinated axons. And outside here, we expect that they would be repaired by central nervous system or dendroglia. However, in here, what has been observed is that the axons are remyelinated by Schwann cells. And my mentor, Dr. Bunke, and I were able to confirm that this happens in people. We conducted an autopsy study of several individuals, and we identified that this phenomenon I've been showing you with the cavitation and the preservation of axons on the outside boundary and the loss of axons on the inside boundary was indeed found in several cases. Further, there was some evidence of spontaneous Schwann cell myelination. So when we talk about doing Schwann cell transplants, we want to utilize this cavity area which is a safe place in general to do a transplant. So we will insert a needle, the Schwann cells are introduced, and what we believe then happens is that those Schwann cells move to the periphery of the cavity and they repair those axons that are demyelinated. In addition, they can form new fascicles of regenerating axons. And this may be more or less effective, depending upon several factors. And when it's really effective, we can see that we're getting quite a good degree of repair on this inner boundary of the intraspinal cord. So let's now talk about reconstructive tissue bridging. So we did experiments for many years at the Univ project where we fully transected the spinal cord, and then we inserted these Schwann cell grafts. And these were soft, they were partially gelated, and they were very reliable to allow for axon regeneration, which is indicated here. This was very reproducible across multiple investigators. Something really interesting that we have observed is that we also get astrocyte processes of the CNS growing into these grafts, which tells us that this tissue can become a hybrid of the peripheral and central nervous system. And so this is a form of integration which we think would be favorable. And this effect was also reproduced in another study by Ryan Williams. And what he was reporting here is that the extent of axon ingrowth from the brainstem was related to how many of the processes of astrocytes go in. So further, we think that this hybridization of the tissue is more supportive of axonal regeneration. And so using electron micrographs, we can look at these fascicles. And so recall that I was saying that we are technically outside of the spinal cord and that we are always going to be compartmentalized. And so you can see these compartments being formed by these fibroblasts, and we have Schwann cells, axons, and astrocyte processes all within one structure. What about new axonal synaptic connections? So we were able to document across several studies that many of the intrinsic neurons of the spinal cord can respond to these grafts. So we think of these as propriospinal neurons and pathways. In addition, many brainstem neurons, which are important for locomotion and posture, also respond to these grafts. However, the corticospinal system does not respond very well to these grafts. So this is the problem that we face, is that we get lots of regeneration into the graft, but relatively little regeneration out of the graft into the distal spinal cord. And we call this the off-ramp problem. And so this has been very reproducible, again, shown by many investigators. And so we have focused on trying to increase this regeneration back into the spinal cord using a variety of techniques such as neurotrophin delivery or enzymes that can degrade the astrocytic scar. So to summarize this part, Schwann cell grafts are very effective to support axonal responses. Re-entry into the spinal cord is limited, myelin repair can occur, and these effects are very reproducible across models and investigators. Let's talk a little bit about clinical transplantation now. So that, as is known to people attending this meeting, there are various challenges associated with initiating and sustaining clinical transplantation programs. This includes cost, regulatory concerns, technical issues, and then the clinical population, which has a lot of heterogeneity in terms of how they will respond to these implants. The assessments that we have often shed limited insight for the mechanisms of action of the cells, and control groups are typically very limited in cell transplantation studies. So this is our paradigm of autologous transplantation, taking a small nerve and then culturing it and reintroducing it, in particular to spinal cord injuries. We've scaled this up over several decades, and now it's a very reproducible paradigm whereby we can create large populations of purified cells for implantation. So that brings us to the importance of cellular suspensions, which are the product of the cell culturing. So this is important because we can optimize the cells during the time that they're in cell culture and transduce them into a repair phenotype. The cellular suspension, when injected, conforms to the injury geometry, and we get multifunctional effects of the cells, trophic, myelin, and regeneration. As well, we can bank these cellular suspensions in terms of when we want to use them in individuals, and we also have now regulatory pathways in place. And then, of course, we have to think about whether we're going to be doing autologous or allogeneic transplantation. For human Schwann cell work, we have not tested allogeneic Schwann cell transplantation as yet. So this is what optimally we get with the Schwann cell implant. You can see the tissue bridges that I was talking about, and the cavity is now filled with Schwann cells. So this is a more or less ideal result. And then we had to develop the injection methodologies to deliver the cells. And so we worked with the syringe positioning device that was loaned to us by Dr. Ed Worth. We used this in both of our clinical trials of Schwann cell transplantation. Furthermore, after we made the transplants, we're able to see them using ultrasound. So this confirms that the cells are indeed deposited into the spinal cord, and we can even see them moving along a pressure gradient. We were able also to transplant into these cavities that I've been talking about, and so this was not an insurmountable obstacle in our clinical trial. So I'm going to talk about some selected clinical outcomes. Much of this is published, and you are free to go and read this. But I wanted to talk about the longitudinal MRI imaging, because this is extremely important in terms of understanding what the implant is doing. So you can see up here, there's a cavitated spinal cord. You can see the dark rim of the spinal cord. And then these studies go over a 1-year period. So you can see that after implantation, the cavity size is reduced, and we see some gray signal inside here. And then we see this additionally at the 1-year time point. We really don't know what this tissue is that's inside the spinal cord as to whether it's de novo tissue or it's extracellular matrix. And that's one of the things that we really need to solve going forward, is to link what's happening after the implant to the actual mechanisms of the therapy. We would like to apply some advanced neuroimaging methods. We're particularly interested in 18F foraminopyridine, because this can actually serve to identify demyelination in the spinal cord, and hopefully it would allow us to see if the cells are mediating remyelination. In addition, we did extensive neurophysiological studies in our clinical trials, and so this is just one example. So at the pre-transplant time point, there were no motor evoked potentials induced by stimulating the brain and recording from the leg muscle of the person. However, 6 months post-transplant, we were able to identify reproducible potentials in a muscle of the foot. Now, we didn't have a control group specifically for this, so we don't know for sure whether this was a natural recovery or mediated by the cells, but it was encouraging. Lessons learned in future directions. So last year, we published a paper in which we summarized our experience and the main lessons that we've learned to this point. So as I mentioned, we have a problem of cell death. The cells, when they are released from the culture, they can initiate cell death programs, and so we can lose a lot of the cells that we place into the graft, and we're working on methods to reduce this cell loss. Furthermore, I mentioned that although the axons grow readily into the Schwann cell graft, they don't leave the graft very well. And so we're working on methods to improve the integration of the Schwann cell transplant into the central nervous system. As I've indicated to you already with electron micrographs, we're very interested in how we can make this hybridization of the Schwann cell fascicle and the astrocyte processes more favorable. So summary of our current strengths and limitations: robust myelin formation and regenerative effects, reliable manufacturing that's FDA compliant, an extremely robust safety record both in preclinical and clinical studies, and then our issues that we're up against are cell survival post-implantation, limited migration, and variable integration into the host tissues. So there's been evolution of these Schwann cell therapeutics, which is one of the benefits of going forward with a translational program. So what we didn't know when we started out is that Schwann cells produce abundant exosomes. And exosomes are powerful signaling molecules that can actually have potent therapeutic effects. And so we've now been able to use exactly the same methods that we used to prepare the cells to produce highly concentrated exosomal preparations. And these preparations can actually be delivered intravenously into individuals. So that's a really remarkable thing. We start out with a peripheral nerve, culture Schwann cells, create exosomes, and then this can actually be an intravenous formulation. So we've been testing this in neurotrauma models, and we conducted one small clinical study in ALS. Important steps for future research: optimize the timing of cell transplantation, improve cell survival, find methods to link what the transplanted cells are doing to mechanisms of action, such as myelination, and combinations with other therapies to potentiate effects. Didn't have time to mention it, but we've conducted some studies that are combined with Schwann cells and gene therapy to express chondroitinase, and those results are very promising. So thank you very much. These are my acknowledgements for the colleagues and students that were really critical for getting our part of the work going, and also the funding organizations who allowed both the preclinical and clinical translation to occur. Thank you very much.